So most people in this room have heard of the ALS Ice Bucket Challenge, but how many of you actually know what ALS is? Often the people who know the disease best are the patients themselves. So imagine you've all just been diagnosed with ALS, or as your doctor calls it, a myotrophic lateral sclerosis. You've been told you have 3 years to live, and over the next 3 years, your nerve cells will slowly die. First, you'll lose the ability to move, then the ability to speak, and finally, the ability to breathe. And what's worse, there's no cure. The most you can hope for from drugs is that they might merely slow the inevitable progression of this disease. And why is this? To be honest, it's because we as scientists don't know enough about ALS. But this is where my research comes in, because I believe that with the correct combination of biology, physics, and chemistry, we can fill in the information in this gap. So let's start with what we know. We know that the human body contains billions of nerve cells, and within each of these nerve cells, there are millions of tiny little machines called proteins. And these proteins function every day to make your life possible. But in ALS, one of these proteins is misbehaving. And this brings us to what we don't know. We don't know why this protein is misbehaving. And in biology, shape is very closely correlated with function, like in everyday life. So this should be a pretty simple question, because if I can look at the protein, I can understand why it's misbehaving. But remember, we've just zoomed into our bodies over 1 trillion times. We're now at a level where microscopes don't give me any useful information. So this is where I bring in some physics tricks. Similar to the way your doctor might use the physics of magnets and X-rays to get structural information about your body, as you can see in these MRI images and X-ray images, I used the physics of magnets and X-rays to get structural information about my proteins. Because if I know what these proteins look like, I can start to predict their behavior. And if I understand their behavior, I can start predicting what small molecules in chemistry might inhibit this misbehavior. So every day, I work at the intersection between the biology of cells, the physics of magnets, and the chemistry of small molecules in hopes that one day I can help contribute to finding a cure for this horrible disease. Thank you.